Ever notice how the coolest new medicines don't bother knocking at the cell's front door anymore? They just slip right inside, head for the control panel, and start rewiring the place. Pretty wild, right? So the real question becomes, once you're inside, where do you aim next? Think of older drugs as polite locksmiths. They fiddled with the door lock, the membrane receptor, hoping the room behind it would calm down. Today's researchers? They stride past the foyer and head straight for the breaker box where all the real decisions get made. Inside that control room is a maze of switches, dimmers, and circuit breakers, deciding when a cell should grow, divide, or call it quits. For years, we called most of those switches undruggable. No handy pockets for small molecules, plus they hide deep in the cytosol or nucleus, places we thought were off limits. Well, that idea is officially outdated. Thanks to better chemistry, sharper structural snapshots, and smarter delivery tech, we can now reach those inner switches and fine-tune them. Sometimes we dial a pathway down instead of killing it. Other times we reroute the whole circuit. So, first big signpost. Drug discovery is no longer just, which receptor do we block? It's, how does the entire signaling route behave, and where's the one choke point that brings the network back to normal? It's like tracing a city's power grid to the substation, not just flipping the nearest light switch. Here's the surprise from those maps. The real action is mostly away from the membrane. 2/3 of our proteins live in the cytosol or nucleus. Picture kinases passing phosphate batons, phosphatases grabbing them back, small G proteins toggling on and off, adapters forming pop-up teams, transcription factors hauling messages into the genome. It's rush hour traffic in there. Now, let's talk tools. Only 3, so nobody's brain melts. First, classic small molecules. They still shine at slipping into deep pockets and shutting enzymes down, but we've upgraded them. Allosteric modulators bind at a side site and act like a dimmer switch instead of an on-off breaker. Second, targeted degraders, PROTACs. Different philosophy here. Rather than block the troublemaker, they slap on a removal tag. One quick handshake and the protein heads to the cellular trash compactor. A fleeting encounter turns into a long-term cleanup. Third, RNA-based therapies. They step even earlier in the information chain. By lowering, or editing, the amount of a signaling protein the cell makes, they trim an overactive pathway up at the blueprint stage. No folding needed, it's programmable code. Let that sink in for a moment. Because we can mix and match these tactics, our view of risk and selectivity is changing. Instead of fearing any move inside the cell, scientists now dissect branches and feedback loops, looking for that one traffic light that slows disease-only lanes while letting healthy routes cruise. Here's a quick analogy to glue it in place. Picture a project team. 1. A kinase blocker is the strict project manager. You, stop talking. 2. An allosteric modulator is the coach. Lower your voice, collaborate better. 3. A PROTAC calls HR. You're reassigned elsewhere. 4. An RNA drug handles hiring. We just won't fill that problematic role. Different diseases need different HR policies. An aggressive cancer might call for the HR route, total removal of a mutant hub. Autoimmune flare-ups? Maybe a gentle coaching tweak is safer. And combinations? Oh, they're looking powerful, like lowering a pathway's baseline with RNA, then fine-tuning the moment-to-moment action with an allosteric touch. Fast forward a few years. We may treat each person's signaling landscape like live weather, not a fixed wiring diagram. Clinicians could tune intracellular targets the way a sound engineer balances a concert. Short bursts of precise modulation, then recovery windows to keep the music clear without blowing the speakers. What does that future require? Less about one magic bullet, more about orchestration, layered, time-staggered nudges that keep the network resilient while steering it away from disease. Imagine remixing a playlist instead of blasting one loud track all night. Before we wrap, I've got a challenge for you. Without peeking at your notes, can you explain the difference between targeting a kinase and a phosphatase? If that feels fuzzy, there's your study topic. Next, pick a pathway you care about, maybe PI3K, AKT, mTOR, or JAK-STAT. Which node looks druggable to you and why? Is it the structural pocket, an allosteric site, a clean biomarker? Finally, if you had to pitch one intracellular protein as a first-in-class drug target today, what 2 or 3 data points would you grab? Structure, pathway maps, disease linkage, and where would you find them? So here's what we covered today. 1. The drug world has moved past the cell's front door and into the control room. 2. We've got a trio of new tools: upgraded small molecules, protein degraders, and RNA therapies that let us dim, delete, or downstaff intracellular troublemakers. 3. Success now hinges on orchestration, not single shots. Think layered nudges tuned over time. And 4. Your mission is to spot the most promising choke point in your favorite pathway and back it up with tight data. If you can do that, you're already thinking like the next wave of drug developers. Go map that control room, and we'll see you next week.